Hi, my name is Adithya Madduri, and I'm currently a senior at Maryville High School. Over the summer, I had the honor of serving as a Borlaug-Ruan intern at the World Vegetable Center in Taiwan under the mentorship of Dr. Hamid Kazai. For my internship, I chose to focus on the potential of wild relatives of tomatoes and their role in breeding for biotic stresses. The goal of my internship was twofold. First, to understand and explore the major diseases facing tomato crops, and secondly, to find genes for disease resistance that could then be used to confer disease resistance to cultivated tomato crops. In order to confer disease resistance, scientists normally use a process known as plant hybridization. Plant hybridization is the process of crossbreeding between genetically dissimilar parents to produce a hybrid. Through this process, scientists have combined a variety of wild-type crops to create hybrids that have higher fitness than the wild type alone. In order to create these hybrids, scientists sometimes use introgression, which is the transfer of genetic information from one species to another. Past research has shown that crop wild relatives and their use in hybridization and introgression have significantly enhanced agricultural production by introducing beneficial characteristics to the breeding pool, and I therefore wanted to explore how they might be used in the context of my own internship in order to prevent disease in cultivated tomato crops. Tomatoes were an excellent species for such wild-type research because they have a wide genetic diversity. Tomatoes are native to South America, and all modern-day species of tomato are descended from the region. The genetic diversity of tomatoes is reinforced by the fact that scientists have recently discovered two new species of tomatoes near Chile and Peru. Due to this wide genetic diversity, it was necessary for me to review the vast amount of available literature on different species of tomatoes, and I used this information to compile the first part of my report. Next, I analyzed the diseases facing tomato crops across the world. For each disease, I plotted the environmental and climatic conditions where it was most prevalent and examined the symptoms associated with each disease. I not only compiled a chart for my report, but also compiled a map of all the diseases so that I could use it during my presentations. After that, I investigated the various wild species of tomatoes, such as Solanum chilense and Solanum pinelli, to see which genes these plants had. From this, Dr. Hamid and I linked resistance genes with specific diseases. After that, I compiled the rest of my report on the use of these genes in order to prevent disease. During my research, I also discovered that though there are many benefits to plant hybridization, there are also certain drawbacks. One drawback was linkage drag, wherein a mixture of two wild types results in the negative traits of both species being amplified. This often creates tomato crops that look nothing like normally cultivated tomatoes and are unsellable for most farmers. In order to prevent linkage drag, scientists have used many new tools. Two of the tools that I decided to focus on were quantitative trait loci and the use of gene mapping. Both of these tools have allowed scientists to combat the drawbacks of linkage drag and have therefore allowed for more successful and vigorous hybrids. To conclude my internship, I submitted a final report that detailed the incredible potential of wild-type breeding in order to combat the diseases found in tomato crops. Overall, through the course of my internship, I was able to explore my passion for biology and environmental science and see what a career in those fields might look like. I've come away with a greater appreciation for scientific research and desire to conduct more research on my own. Before I conclude, I would like to thank everyone who made this internship possible. From my AP Environmental Science teacher, Mr. Jones, to my family, both of whom pushed me to apply for this internship and both of whom have inspired my interest in science. I would also like to thank Ms. Rebecca Picard, who coordinated my internship with the World Vegetable Center, and I would of course like to thank Dr. Hamid Kazai, who continually offered his support and generous advice over the course of my internship. Thank you for this incredible opportunity